we're on our about third generation of spatial light modulator system now, and our latest release is called the NeuraLight 3 Ultra, where we've increased the field of view of our spatial light modulator, so you're able to activate cells that are kind of farther apart in your sample than you could before. This custom chip that we did also allowed us to increase the resolution, and so their stimulation resolution is improved by a factor of 2, where we're now able to more precisely activate certain cells without accidentally activating cells that you didn't intend to activate. This is also the fastest spatial light modulator on the market, which allows us to change the pattern of stimulation at up to 600 patterns per second, so that you can stimulate one set of cells and move on to the next set of cells pretty quickly. And we couple this with our ETL, which we'll get into, which allows us to do 3D imaging at the same time as the stimulation. And so we're really grateful to our collaborators at Columbia University, Stanford University, UCL, University of Oxford for this great collaboration to bring this to market. And what I showed on the previous slide was just 2D activation of cells while doing calcium imaging. But this slide shows the ability to do 3D activation of cells in your tissue as well. And so this is data from Dr. Ahmed Arak, who beautiful data that shows that you can do calcium imaging and activate. What he's showing is 3 planes, many cells at once, separated by, in this case, 20 microns apart in the cortex. We can go a lot deeper as well and a lot thicker, but this is great looking data from Professor Arak's team at UCLA. And our contribution to this effort was really a lot on the workflow side of things. And so our goal is to try to create software and the workflow that allowed for users that may not have the optical capability to build their own spatial light modulator in their labs to be able to do these sorts of experiments. And so over years over the 10 years that we've tried to refine the software, we reduced the complexity of the software essentially down to the user needs to place down markers of where they want to stimulate or the cells they want to identify to stimulate, define the amount of time they want to stimulate, the laser power, essentially click go, right, and start the imaging. And so we wanted the user to be able to focus on the biology and the expression levels of your calcium indicator, your opsin. That's already difficult enough. We wanted to make the acquisition side of things as simple as possible. And going back to our roots a little bit, we have users that have also used this spatial light modulator and this holographic technology, not to do calcium imaging and opsin activation with optogenetics, but to do 3D glutamate uncaging. And so this is work from Krishna Jayant's lab at Purdue University, where he published this in Cell Reports, where he's able to do glutamate uncaging at spines across different branches of dendrites, and doing this with high precision with the spatial light modulator. And so with a lot of behavioral experiments and calcium imaging and neural activity experiments, sometimes you don't know which cells to activate before you start your experiment. And so this is a request particularly from Michael Hauser's lab and others, where they wanted us to give their lab, a hook into our system so that they're able to do this in real time and in a closed loop fashion. And what I mean by that is that they're doing calcium imaging and they want to identify the cells that activate in real time through their analysis pipeline. And so we have this open programming interface, which we call Prairie Link, that allows them to get the data in real time, put it into their programming language, whether it be MATLAB or Python, where they do their analysis and identify which cells they want to activate, and then send back a command through their program to say, stimulate this cell in X, Y, and Z, or change laser power, or move the stage. And so they can do this all in closed loop fashion. And with the advent of development of AI in recent years, we've also started experimenting with AI for use in this analysis, where the way that we've implemented AI is that we've created a framework where it can host your AI model, as long as it's an NVIDIA engine, into our system, where it can be used to process the data in real time. And so this example is from 𝘃𝗶𝗱𝗲, which was published in the last couple of years, which is, in our hands, a very cool model that allows for denoising of microscopy data in real time on a NVIDIA GPU. And it doesn't have to be this model. You can plug in another model that you train. And Prairie View, which is our software, allows you to denoise or use AI for any and use it for other sorts of analysis.